In this lecture, we will look at how the kidneys buffer. When we looked at buffering, we had chemical, which were the first. They worked in seconds to minutes. We had our respiratory, which are the second ones to jump in. They only work if our lungs are functional and they kick in minutes. And then our final buffer is actually our renal buffers. And these are the best ones when we look at how the kidneys buffer or compensate, because the kidneys actually control the secretion of both bicarb and hydrogen. So they could actually clear it and get it out of your body, which is critical. We know we generate acids every single day. You don't need to memorize the number of milliequivalents, but every day our kidneys have to clear and get these acids out of our bodies. So they're critical for that. And then they also help if our CO2 levels should go up. So CO2 can enter the renal tubules, and CO2 can get into the fluid. And when that CO2 goes in, it undergoes the reaction you just saw to generate hydrogen and bicarb, and the kidneys help eliminate that. Or if you have the lungs are the problem and you have an issue with CO2 levels and you're having changes to your pH, the kidneys can jump in and they can help buffer that. So the first thing to understand is that if we're looking at our DCT cells, here's my DCT, here's my filtrate, here's the blood and the peritubular capillaries. I want you to understand what happens is that CO2 can leave the blood. So maybe your CO2 levels are elevated because of a lung issue, COPD. They go into the tubule cell where they do the same equation. They combine with water and form carbonic acid. And then we get a hydrogen and a bicarb. Now that bicarb can go right back in the blood where it can be used to buffer. That's pretty important. This hydrogen though is going to leave and go out into the filtrate, where it will have to get buffered as it leaves your body through your urine. And some of that is also buffered by bicarb. And so what we know is that every time we bring either a hydrogen back into the blood, that we have this one-to-one relationship with sodium. So most sodium is going to leave the tubule fluid and go all the way back to the blood. And then we know that our hydrogen can be pumped out. But we also know that this reaction, this CO2, if it gets into the filtrate and it goes through the reaction and forms your hydrogen and your bicarb, that any bicarb that's out in the filtrate is stuck. It's trapped. It can't get back inside the tubule cell. So here's another picture showing you. Here's my tubule cell, my extracellular fluid. See my bicarb and chloride going in different directions, and you can see my sodium and hydrogen pumps. And so what happens though is any bicarb that ends out here is trapped. It will never come back through. This side of the tubule cell is impermeable to bicarb. It can only come back on the other side, which is actually good because these hydrogen ions need to be buffered. And so they'll be buffered in the filtrate with the bicarb. So that's a common way that they're buffered. They're also buffered in the filtrate with the ammonia. We talked about the amino group can combine with a hydrogen to form ammonia. So we do have some of this out in our filtrate, and that can also combine with a hydrogen. So NH3 plus a hydrogen forms what's called ammonium, NH4, and that's what we can excrete in our urine as well. So we do have to buffer hydrogen ions out here. When you eliminate this ammonium, a lot of times it attaches to a chloride, so it's called ammonium chloride. And if you've ever noticed if urine sits for a while, it can smell really, really bad. That's because the ammonium, NH4, converts back into ammonia, which is NH3, which has that obnoxious smell. So when we're looking at acidosis and alkalosis, the kidneys can directly eliminate hydrogen or bicarb. We know that hydrogen and potassium compete for secretion. So again, I'll just throw it on here once. We know that sodium's coming back, and that when it comes back, that we're going to have this condition where we can do something with our hydrogen and our bicarb. So what are we going to do? Well, let's say we're in acidosis. So there's some sort of acidosis that occurs. And if we're in acidosis, our blood, then we know that if we're in acidosis, that we have too many hydrogen ions. So if we have too many hydrogen ions, we're going to want to get rid of them. So we're going to increase secretion of hydrogen. We're going to get rid of the hydrogen, but that means we're going to hold on to potassium because they can't both go. And so we're actually going to retain more potassium. So when we're in acidosis, we might be a little concerned about hyperkalemia. Our potassium levels might start to go up and could be a problem. We also know if we're in acidosis that there's two things the kidneys do. We have to look at what they do to hydrogen, and then we have to look at what they do to bicarb. So we are in acidosis, we want to hold on to the bicarb. We want that in our blood because that's our base. It's our very important base. And in this case, the bicarb comes back and we're going to get rid of the chloride. So acidosis directly results in an increased secretion of hydrogen and a decreased secretion of bicarb, so we can restore pH homeostasis in the blood. What if it's alkalosis? Okay, well, if we have alkalosis in our blood, now we have too few of those hydrogen ions in our blood. So now we want to hold on to them. So we will not eliminate them. We're going to kick out potassium. So we would decrease secretion of hydrogen. We're going to hold on to hydrogen, but we're going to get rid of potassium. Well, we could start losing too much potassium, which is easy to do, and end up in a hypokalemia condition. When we're in an alkalosis state, we don't want to hold on to bicarb. That's our base. We're going to get rid of it, but now we're going to retain chloride. So the kidneys directly control the secretion of hydrogen and bicarb in response to the blood pH to restore homeostasis. This is a screenshot from a PhysioX lab that we're not doing. It's helpful if you want to do it, you can, but I just want to take you through this for a minute because it's showing you the response from the kidney for acidosis and alkalosis. So you have your nephron here, which should look familiar from your other lab. And what we do in this lab is you can change the CO2 levels and the blood pH, and you can measure what happens in your urine. So I'd like you to understand this or run through the lab. So we have a normal blood pH, 7.38. Remember, our normal range for arterial blood is 7.35 to 7.45. And my normal pCO2 levels are going to be 35 to 45. So when we have normal CO2 levels, normal pH, we have our normal amount of hydrogen and bicarb. Notice now, though, my pCO2 goes down. Well, we said last lecture that if there's a problem in my lungs, and if my CO2 levels are too low, I'm probably hyperventilating, and my pH in this case is going to become more alkaline. So when my blood is alkaline, notice my urine, I will decrease my secretion of hydrogen. I'm going to hold on to that, but notice I'm going to increase the bicarb in my urine, which is what we just said on the previous slide. Or what if I'm in respiratory acidosis? So my CO2 is too high, I'm not moving the CO2 out. Well, we know if I'm retaining CO2, I'd expect my pH to go down. So I'm in acidosis. Now I'm going to increase my secretion of hydrogen. More hydrogen leaves in the urine, but I'm going to hold on to the bicarb. So I will decrease my secretion of bicarb. And so this is how the kidneys are going to buffer for us, because they directly regulate the hydrogen and the bicarb in the urine to restore the homeostasis. So this wraps up our lecture on kidney acid-base balance and buffering.